The science behind each phase. Let's get into episode 9 of the cell culture series, Thawing, Seeding and Passaging, the complete cell life cycle. From a frozen cryovule to a healthy, expanding culture, every successful cell follows the same journey. Today, we'll walk through each step of that life cycle and learn the techniques that every cell culture scientist must master. Your cells arrive frozen in a tiny vial the size of a lipstick cap. 12 hours later, they can be dividing happily in a flask. But one wrong move between those two moments and they are all dead. Today we are going to discuss the complete workflow from frozen vial to thriving culture. From vial to flask, everything can go wrong. A frozen cryovule contains cells in suspended animation. Thawing wakes them up, seeding gives them a home, passaging keeps them going. These three processes together define the life cycle of every cell culture experiment. Each has specific critical steps, and each has failure modes that are irreversible once triggered. Why this episode connects to everything. Every technique you will ever use in cell culture begins with cells that were properly thawed, seeded at the right density, and passaged at the right time. Errors here do not just affect the current flask, they affect every experiment built on those cells. Now let's check how well you remember one of the most important concepts from our cryopreservation episode. Here's your question. Why must you dilute DMSO from the thawed cell suspension immediately after thawing, rather than waiting until after counting the cells? Is it A. DMSO interferes with cell counting accuracy? B. DMSO becomes increasingly toxic as temperature rises, so every minute of delay at 37 degrees Celsius reduces cell viability? C. DMSO causes cells to aggregate, making counting impossible? Or D. Immediate dilution is not necessary because DMSO is only toxic during the freezing step? Take a moment to think about your answer. The correct answer is B. As we learned in episode 6, DMSO protects cells during freezing, but once the cells are thawed and warmed, it quickly becomes toxic. As metabolism resumes, DMSO starts damaging the cells, and every minute they remain in warm DMSO reduces their survival. That's why the correct workflow is simple. Thaw the cells, immediately dilute the DMSO with warm culture medium, and then proceed with cell counting. A delay of just 5 to 10 minutes can reduce cell viability dramatically, from around 85% to nearly 40%. So always remember this rule: dilute first, count second, your cells will thank you for it. The science behind each phase. Phase 1. Thawing, recap plus extended protocol. Rapid thaw in 37 degrees Celsius water bath, 60 to 90 seconds, immediate dropwise dilution into pre-warmed media, centrifuge to remove DMSO, replate. We covered this in episode 6, but here we add the nuances specific to different cell types and the post-thaw recovery period. The 24-hour recovery rule. After thawing, cells need 24 to 48 hours to recover from cryogenic stress before you use them in any experiment. Check them by phase contrast after 24 hours. If morphology looks stressed, give them another 24 hours. Starting an experiment with cells still in DMSO stress recovery is the same as starting a race on injured legs.